The overall goal of this procedure is to isolate and purify subsets of human blood vessel-derived multipotent precursor cells from a single skeletal muscle biopsy. First, dissociate the muscle biopsy and digest with a combination of collagenase type 1, 2, and 4. Then, label the single-cell suspension with fluorescence-conjugated antibodies that recognize a combination of select cell surface markers. Next, perform fluorescence-activated cell sorting by flow cytometry. See and expand the sorted cell subtypes in specific culture conditions at optimal densities. Ultimately, the post-sort analysis of purified cells, as well as subsequent flow cytometry and RT-PCR analysis of cultured cell subsets, is used to show the purity of these homogeneous BVSC subsets.
Yeah, this method, can answer really, what is the origin of those mesenchymal stem cells, because a lot of people have found different type of stem cells in different tissues. So I think this can answer the origin of those stem cells, as well as the role that perivascular and any other blood vessel-derived cells play in tissue regeneration and repair.
Visual demonstration of this method is critical as the muscle biopsy dissociation steps can be difficult to learn. Technical details and optimization of collagenase digestion are very critical to the yield and the purification of target cells.
Transport the human skeletal muscle biopsy on ice.